Okay, so like I said, we're going to take these last three sections in this chapter kind of slow. And the reason we're going to take them kind of slow is because there's a lot of detailing here that if we just gloss over it, we're going to run into some problems with. And so we're going to get a little bit of context today. We're going to kind of set the table up a little bit. And then tomorrow, I'm going to give you an interactive to kind of play around with. That'll kind of help visualize and activate these processes a little bit better. And then we'll talk about mutations, which is when kind of these processes go wrong and we see some things that we're not supposed to see. So the primary topic today is RNA. Now, we've talked about RNA from a structural standpoint. Kind of two big differences between DNA and RNA. RNA, we have the ribose sugar as the sugar backbone instead of deoxyribose. Again, remember the difference there is just the presence or absence of an OH group on the second carbon in that ring. The other difference between DNA and RNA is the presence of this base group uracil. And the fact that the uracil stands in the place of thymine, where thymine would be used in DNA. Now, another important difference in RNA compared to DNA, RNA is usually a single strand. And the reason why it's a single strand is because of what its job is. Now, we talked about DNA. DNA's got the double helix. It's got basically the two strands working in opposite directions. And they're being connected to each other by these complementary base pairs. Well, RNA's primary job is to do the work of taking that DNA and making something of it. And so it doesn't need to be a double helix. It doesn't even need to be double stranded, because basically its job is going to be, it's going to copy half of the genetic code and then use that genetic code to create amino acid groups and proteins. And we'll get more into that later on here. So there are three primary types of RNA inside of cells. The first kind of RNA is called messenger RNA. Messenger RNA, mRNA. This is the version of RNA that actually goes into the nucleus of the cell and copies the genetic information. So let's say that you've got a gene and you want that gene to be transformed into some sort of functional protein. Well, what happens is a series of processes happen inside of the nucleus of your cell to copy that particular part of the DNA and turn that DNA into something that can be used. Why it exists has a lot to do with the fact of size. Inside of your nucleus, there are a bunch of chromosomes. Those chromosomes are actually large enough that if we have a good microscope, we can actually see them. And inside of each of those chromosomes, you have tightly wound DNA strands, bunches of them. Now, the individual DNA strands, we can't see with a normal microscope. We usually need something a little bit more powerful, like a scanning electron microscope to do. Problem is, if I'm interested in creating a protein, I don't need the whole chromosome. I don't even need the whole DNA. I need a small portion of it. And that big chromosome, it's going to be hard to transport inside and out of the nucleus. It's kind of a big molecule. It's not going to fit through those little tiny holes that can hold water. So what the RNA does is it's able to go in through the use of an enzyme, snip off a portion of the DNA that's relevant, and because it's smaller, it can fit through one of those little membrane holes and get to the outside of the nucleus. And once it's outside the nucleus, then this other stuff can take over in terms of making a protein. And so that's where transfer RNA comes in. Transfer RNA takes a look at the messenger RNA and starts to pick up amino acids based upon coding information. And we'll get into the codes, here in a second. But what it does is it allows us to create these amino acid chains that will eventually become a protein. Ribosomal RNA, this one we won't talk about nearly as much. This is actually the most common type of RNA. This is the stuff that is sitting around in your cytoplasm. And when we combine this RNA with the proteins, they make something called a ribosome. And the reason the ribosomes are so important is because these are the sites where protein synthesis actually takes place. So we'll start with transfer RNA, tRNA. And tRNA has a look like this. Now, this is actually, believe it or not, this is actually a single strand. What we have to hold this particular shape is a series of hydrogen bonds to help connect some of these groups to each other and create this kind of a three-dimensional shape. Now, not all transfer RNA look exactly the same, but they do have some commonalities. Each one of these has somewhere between 70 and 90 nucleotides in it. On the 3 sugar end, there is a opening sequence, ACC. This is known as the acceptor stem. This is what is going to help attach the amino acid to the protein chain as it is being sequenced. And attached in here is going to be something called an anticodon. Now, this term isn't going to make a whole lot of sense until we talk about codons. But basically, what we have in a codon versus anticodon is we've got a complementary base group. And that complementary base group codes to a specific amino acid. And so again, we'll kind of focus on that when we get more towards the translation side as opposed to just the transcription side. In protein synthesis, there are two processes that happen, and they do not happen simultaneously. They happen in turn. So the first thing that happens is transcription. And in the transcription process, what we have is an enzyme comes inside of the nucleus, opens the DNA up and transcribes, using the complementary bases, transcribes a particular portion of the genetic code. That's what happens in transcription. So we take a portion of DNA called a gene, we copy it over, but unlike the process we talked about at the end of last Tuesday, where we were replicating the DNA strand exactly to create two new strands of DNA, what's happening here is we are just pulling off a portion of it, and we're going to get the messenger RNA as a result. That messenger RNA is a snippet of the genetic code. It's a small strand. It worms its way out of the nucleus into the cytoplasm. And that's where this second process can take place. The second process of translation turns our messenger RNA into amino acids. And once we link together all of the amino acids, that amino acid chain is able to fold on itself and translate into a functional protein. So if we're looking at the two processes, kind of just summarizing them into quick snippets. In transcription, what we are seeing is that messenger RNA is copying the genetic code. So it's taking that DNA, it's transcribing it into a simple code that can leave the nucleus. Translation, on the other hand, we're going to see a conversion process that takes the messenger RNA and turns it into a protein. Now, I'm actually going to jump ahead a little bit here because I think these ideas are super kind of ethereal. They aren't going to make a whole lot of sense until we see it. And so what I've done is I've pulled a couple of pretty good modules, animations, and we're going to focus on this first one today. On tomorrow, we'll look at this second one. Second one's a little bit more interactive for you, but it should help kind of put some of this into context here. So if we're looking at transcription here, Okay, so just as kind of a review here, let's start with just kind of the overview here. We're talking about protein synthesis here. Protein synthesis happens inside of cells. And the instructions for that protein synthesis are found in the DNA. So this is an example of a plant cell, but the processes that occur in these plant cells are very similar to what would occur in your cells as well. Here in the middle, this is our nucleus. The nucleus is the housing of all of the genetic material for a given cell. Every single one of your cells has one of these nuclei. Depending upon the species, you could have many chromosomes, you could have a few chromosomes. We happen to have 23 pairs of chromosomes. Again, that's in every single cell of your body. Inside of each one of those chromosomes, there is your DNA. It is wound up very, very, very tightly in this coil. So not only are we seeing the double helix that we've been talking about all this time, we're seeing that this double helix has a lot of winding and winding and winding to make a very compact structure. If we looked at any given coil here, this is where we would see that double helix that we're the most familiar with. And inside of any one of those double helixes, we would see the complementary base pairs. Now, taking this back all the way, we could see why it would be a problem for these chromosomes to travel in and out of the nucleus. They're quite large. Relative to the cell membrane itself, they're gigantic. Things that go through membranes tend to be small. They tend to be things like water or small ions. Maybe an occasional protein if we have a channel built into the cell membrane that allows proteins to go through. But these chromosomes are gigantic by comparison. And so if we're going to make proteins, we can't do it by having the chromosome leave the nucleus. What we have to do instead is we have to have some way of the chromosome imparting its knowledge to the outside of the cell. And that's where these enzymes come in. So this green ball here represents an enzyme. What the enzyme is going to do is it has straightened out one of the coils of the DNA here, and it's going to transcribe it. It's going to zip its way across here, and using the complementary base pairs, it's going to read the genetic material and create a strand of messenger RNA. So that's what this red ribbon hanging off of this green enzyme is. That's the messenger RNA. Now that messenger RNA because it is so much considerably smaller than the chromosome that it came from, it can travel through that nuclear membrane and get outside of the nucleus. And so that's what transcription is all about. So let's look at transcription a little bit more closely here then. In transcription, just giving us a little bit more fine detail here. We usually find some sort of active site on the DNA code itself that tells the enzyme where to hook on. That's called the promoter. The promoter is the part of the genetic code that turns the gene on or off. Then we have a coding region. This is the area that the messenger RNA is built from. And then there's what is called a termination sequence. And the termination sequence tells the enzyme, okay, stop here. The enzyme in question is something called RNA polymerase. RNA polymerase is what is going to attach itself to the DNA, zip open the DNA, and copy the DNA to create this messenger RNA strand. And so if we look at this just a little bit closer, here's the promoter region. This is the part that is going to tell the gene to come on or come off. The RNA polymerase is going to recognize the promoter based on chemical markers. It's going to attach itself. And when it attaches, look what it does to the DNA. It zips it open. So now that DNA, which was twisted in that double helix, now it has been opened up. And in opening up, once we hit the coding region, now we can start to build a copy of the part of the gene that we're actually interested in. And this is where the messenger RNA starts to be made. As it goes through the coding region, we can see that it's basically just building a copy of the top strand, only substituting in that uracil instead of thymine, because that's what messenger RNA is made from. And so it's going to continue to do this. It's going to continue to unwind and build, unwind and build, unwind and build until it gets to the termination sequence. Termination sequence is the part of the genetic code that says, okay, stop copying and detach. And so the RNA polymerase stops copying. The RNA gets detached. And there's my messenger RNA strand. That messenger RNA strand now can exit the nucleus and head out to the cytoplasm. And in the cytoplasm, that's where the transfer RNA is. That's where the ribosomes are to start building out the protein. Okay. Do the pictures help at all? Okay. So that's the transcription process. Transcription process happens inside of the nucleus of the cell, and that is what is going to convert the DNA into messenger RNA. That's the first step of this process. The second step of the process is we take that messenger RNA and we translate it into making a protein. And how that works is, notice that the messenger RNA here has taken those base groups and put them into triplets. 3 base groups in a row, gap. 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 Well, that's intentional. And the reason why that's intentional is because when we go from the transcription to create that messenger RNA to the translation phase, this 3 base sequence is going to translate to a particular amino acid. And so what the transfer RNA does is it looks at this and says, oh, okay, these 3 base groups, well, that's glycine. These 3 base groups, that's alanine. These 3 base groups, that's isoleucine. And it just picks them up one after another, after another, after another, after another, to make this chain that's going to become our protein. Let's take a minute here and just kind of review what we've done so far. So here I have my DNA strand, and this particular strand is CTA-AGG. What would the complementary messenger RNA strand look like that would match this particular part of the genetic code? The correct answer is B. Remember, since we're talking about complementarity here, that means that complement of C is G. Complement of T is A. Complement of C is G, G is C. The reason A is not correct is because thymine does not exist in RNA. It's uracil instead. So for C, I would need a G. For T, I would need an A. For the A's, I would need U's because there is no thymine. It's uracil instead. And for the G's, I would need C's. That gives us option B here. All right. If we look further into this translation piece, this is the genetic code. Now, this genetic code, like I said, forms in triplets. Those triplets are known as codons. So this is where that codon anticodon thing comes from. These codons in the messenger RNA will have complementary transfer RNA anticodons that tell the transfer RNA which version of transfer RNA needs to attach. So if I have a codon that says UUU, the anticodon there would be AAA. And so I would need to find the transfer RNA that has the anticodon AAA. That would hook up right here. And that would give us a phenylalanine amino acid. So the triplets here give us the codons that tell us which transfer RNA need to attach. And the codons have been determined. They are a 3-base sequence that give us one of the 20 amino acids. Now, what you need to understand, and this is what can be really confusing, obviously, with 3 different positions and 4 different bases per position, there are a total of 64 combinations, or cubed. 64 combinations of 3-letter codes. But yet there are only 20 amino acids. So what that means is that there's a lot of duplicity here. There's a lot of degeneration. Here's an example. So GCA. GCA refers to alanine. Alanine is the amino acid that corresponds to this particular codon. But it's not the only combination that gives us alanine. GCC, GCG, GCU, all 3 of those, in addition to GCA, give us alanine. What's important, though, is that these codons are precise. GCA, GCC, they only code for alanine. They don't code for any of the others. Now, what's also important, just like we saw in the animation, where there is a promotion stage and a termination stage, there are promotion and termination codons as well. So chain initiation will always start with AUG. So that's kind of a marker on the messenger RNA to say this is where you start coding, this is where you start translating. Any one of these 3 combinations, UAA, UAG, UGA, those are all stop codons, termination codons. The other really interesting tidbit is that this genetic code translates to most organisms, if not all organisms. The only reason we can't say all organisms is because we haven't cracked the genetic code of every single organism, plant, and animal that exists. But for the ones that we do know, they all fit. They all fit. So GCA is alanine. Doesn't matter if you're looking at a human being, doesn't matter if you're looking at a tree. Means the same thing. Codes the same amino acid. Genetic code keys like this are what are important for us. Because this is going to tell us how to interpret any 3-letter sequence to figure out what it is. And the way that you read it is from left to top to right. So my first letter is U, my second letter is C, my third letter is A, UCA, that's Siri. And so what this allows to happen inside of your cells is now that your messenger RNA is out in the open, your transfer RNA is able to take that code and bring in the correct amino acids in the correct order. And we start building that chain. One after another, after another, after another. We get to a stop codon, everything detaches, and then the protein's able to fold up on itself and create that three-dimensional activated protein that we would expect. So what we can do with this kind of information is we can answer questions like this. If I have this messenger RNA sequence, what that tells me is, okay, GCC is this one. So alanine would be the first part of the first amino acid. GUA, okay, that's valine. GAC, okay, that's aspartic acid. And then obviously that could go on and on further and further. So where we will pick up tomorrow is, if you usually don't bring a computer with you tomorrow, that's okay. I've got a QR code set up here so you can do it on your phone. But what we're going to look at is I'm going to show you the other animation to kind of walk us through what translation looks like, kind of in pictures as opposed to just words. And then we're going to go through this University of Utah activity together. Give you an opportunity to kind of work with each other, work together on that. And we'll then finish up the chapter talking about what happens when this genetic code gets altered with mutations. So that's what you have to look forward to tomorrow. Have a great day. I'll see you.